Okay, let's go ahead and continue to talk about the ion channels and talk about whether something is excitatory or inhibitory, and by introducing a concept called excitatory postsynaptic potentials versus inhibitory postsynaptic potentials. Okay, so we call those IPSPs or EPSPs. All right, but back to this. What do we have here? Of course, I have my cell here. And since we're talking about neurons, the cell's a neuron. Here I have my chemically gated channel. And again, I like to call it the keyed doorways. Here's my chemical, my neurotransmitter, GABA. And there's actually the keyhole. So GABA is actually going to fit in there, open up the doorway for a specific ion to be able to go inside. Just so happens the ion is chlorine. And since chlorine or chloride ion is negatively charged, then once that goes inside, it's going to cause my millivoltage inside to actually drop. Well, if it drops, then it's actually, remember, it's already negatively charged in here. So if that drops, then it's going to go further away from threshold. Therefore, that's going to prevent my voltage reaching threshold and opening up voltage gated channels, which start my action potential. Anyway, let's look at this little example that they have for us and listen to their explanation.
Synapses. Ions move across cell membranes through chemically gated channels. The gates are opened or closed by neurotransmitters.
Okay, so again here we saw GABA connecting that, opening up that channel. Chloride moving through there, that's going to change the voltage inside since chloride is a negative ion. Remember, we have this potential because we're normally positively charged right outside the ion and negatively charged inside. So if I allow a negative ion to go in, I'm going to become even more negatively charged. And if I stay even more negatively charged, I'm not going to reach the threshold it takes to start the action potential. Of course, the threshold has to be ultimately reached at the axon hillock, but this leads to whether I'm going to be able to generate it or, in this case, inhibit it. Okay, we'll quickly go back over the types of ion channels and where they are found. But what we want to do is start relating the types of channels to the different, the causing of whether it's an excitatory situation or inhibitory situation, and then talked about the difference between rapid postsynaptic potentials and slow postsynaptic potentials. Okay, but what we need to know going into this is about the electrochemical gradient, which again has to do with the charges on either side of the membrane due to the ions that are found outside versus inside and how the ions are flowing back and forth. We need to understand about the receptors. In this case, that we have the chemically gated receptor here that's receiving the neurotransmitter to open them up. And then, of course, we need to understand some really, really important definitions. What is depolarized? What's hyperpolarized? And what's threshold? What's neurotransmitter? What's an enzyme? What's phosphorylation? Well, we know the neurotransmitter is the actual chemical released at the synapse that will bind to the gate to open it or to open it up, basically. Now, what it opens it up to is according to the type of neurotransmitter and the type of receptor. Okay, but we know phosphorylation is when we add a phosphate to something. For instance, we know that ATP breaks off the N-phosphate group, attaches it to something to release the energy. Therefore, it is phosphorylating something. All right, we know enzymes are proteins that lower the energy of activation and cause something to either be put together, broken apart, or rearranged. All right, so those three things we've talked about quite a bit now, and we've kind of introduced when we say that we have a charge on either side, a different charge, a positive charge here and a negative charge here, that is a polar situation. So in the resting membrane potential, we know that we have the negative charge on the inside of the membrane and positive charge on the outside, again, primarily because of all the positive sodium ions out here. And the fact that in the resting membrane potential, the fact that we have a lot of leak channels for potassium, so potassium leaks out, then that keeps that charge negative because I got a bunch of potassium leaking out, not as much sodium leaking in. So if I have a bunch of sodium outside and now I've got a bunch of leak channels that's allowing another positive ion to leak out, then I've got all this positive charge out here versus the negative charge inside. Okay, but we also have some forces that are going to be either attracting the ions in or out, and that force is the actual charge itself. Now, if I have a positive charge outside and a negative charge inside, and let's say sodium is going this direction, if sodium is going inside, even though it would go through its own channel, then the sodium is not only going inside because it's going from high to low, but it's also going inside because it's attracted to that negative charge. Because it's attracted to that negative charge inside. Now, potassium, on the other hand, has a totally different electrochemical gradient. It will want to go outside because there's a high concentration inside, but because there's a negative charge inside and a positive charge outside, that will actually prohibit it from going out as easily. So when we say electrical gradient, we're literally talking about the charge that's either attracting, so that negative charge inside is attracting the sodium inside. The chemical part of the electrochemical gradient is simply diffusion, going from a high concentration to a low concentration. As far as potassium is concerned, it has a much lesser electrochemical gradient, because even though it has a chemical gradient that is trying to force it to go outside, because chemical gradient means diffusion, going from a high to a lower concentration, it has an electrical gradient that's not causing it to go outside, but that's preventing it from going outside. Because if I have a positive charge out here and a negative charge inside, and that's positively charged, that negative charge inside is still preventing that from wanting to go outside. It will be more likely to be attracted more to this. So I have two forces pushing sodium in, the electrical force, or the attraction towards the negative charge, and the chemical force, which is diffusion, going from a high to a low. And here, though, I have one force wanting it to go out, that's the chemical force, but I have another force opposing it from going out, and that's the electrical force. Therefore, we will always have more sodium coming into a cell than potassium going out based on the electrochemical gradient. But the other factor is that if I have more leak channels for potassium than sodium, then that gives another force, that gives another way of potassium to be able to leave. That's why we always have more, when we have a resting membrane potential that's negative, that has a negative number, that's because we always have a bunch of leak channels allowing a lot of potassium to leak out, not so many leak channels allowing sodium to leak in. Now, I know that was a lot to say at one time. I'm going to give you some videos to watch to demonstrate that, but hopefully you can follow it up to this point. Okay, so depolarizing is when I change that charge inside from a negative charge to a positive charge. Why? Because a bunch of a positive ion rushed in. Hyperpolarization is when I take that number below my resting membrane potential, as we will see. And of course, threshold is the voltage I have to reach to trigger the action potential. All right, a lot of information for just one page, but we'll be looking at these factors. Okay, this very important concept we talked about last video, but I'm going to go ahead and repeat it. It's that important. We find these leak channels, also called non-gated channels. We find them everywhere because they're responsible for that resting membrane potential that occurs everywhere. Again, they're everywhere. They're responsible for the resting membrane potential. Our chemically gated channels, meaning the chemical being the neurotransmitter, are going to be found in the receptive areas where we're going to receive the neurotransmitters, where the synapse is going to take place to this neuron. Well, we find that on our dendrites and on the cell body. So we find these areas here on the dendrites and cell body because that's where they're going to receive the neurotransmitters. So the chemically gated channels, whereas the non-gated or leak channels are responsible for the resting membrane potential, the chemically gated channels are responsible for the synaptic potential, the potential that's caused where the synapse takes place. We'll be talking more about what we mean by that. Okay, and finally, our voltage gated channels, are found starting on the axon hillock and throughout the rest of the axon where it is where unmyelinated or non-myelinated. Wouldn't make any difference to have channels under something where ions can't move through them. So if it's unmyelinated axon, meaning there's no myelin on it, we would find them the whole length of the axon. But if it's myelinated, we would just find it at the nodes, so the channel can open up and allow the sodium in and the potassium out just in the nodes. And of course, since they're voltage gated channel, we have to reach the voltage that opens them up. Well, we call the voltage that opens them up, we call that threshold, and threshold initially has to be reached at the axon hillock. What causes it to be be reached is what's happening with our chemically gated channels. Either they are allowing positive ions to come in all over where the synapses are taking place, and when I add all that up, if it reaches threshold, then we open up the voltage gated channels. If I inhibit that ability, then I will never reach the voltage called threshold, therefore I will not send the action potential. So again, remember, it's just as important to inhibit as it is to excite. It's doing it at the appropriate time is what counts. Okay, so far when we've talked about the neurotransmitters going through the keyhole and open up the doorway, we've been talking about something that is acting directly. Something that's acting directly, okay, but which is usually going to be create a fast ability of ions to flow through, because it's like going to your door, putting your key in and opening it up and you walking through, okay? But we need to understand that there's also an indirect action where the neurotransmitter goes to a doorway, but instead of opening up directly, it sends a cascade of events, triggering several events that eventually opens up a doorway. So again, we see acetylcholine opening up the doorway and then seeing that the sodium rushes in, even though we may have a little bit of potassium leave. Now again, sodium rushes in due to this electrochemical gradient that I was describing. The fact that I have a higher concentration here than here, so it's going to go from high to low. That's the chemical gradient. The fact that I have a negative charge on the inside of the membrane compared to the positive charge here will also attract the sodium in. Potassium can also leave because there's a higher concentration of potassium inside than outside for the cells, even though in this picture it may not be that way, but in cells that's the case. So the chemical gradient is the fact that potassium is going to leave to go from the higher concentration to the lesser concentration. But the electrical gradient is actually prohibiting it leaving, because since potassium is positively charged, if I have a negative charge here, then remember, negative and positive attract each other. So instead of this potassium going out, that negative charge on the inside is actually trying to keep it in the cell. So I'll never have as much potassium leaving as sodium coming through the same channel. Okay, here we see another direct acting neurotransmitter on a chemically gated channel here. And
we have shown only a single.
Okay. And here, so acetylcholine was direct acting, GABA is direct acting, glutamate is direct acting, glycine is direct acting, meaning they directly open the doorway, which allows the ions to either go in, go out, or positive or negative directly through the doorway. Okay, now notice we're looking at what we call rapid excitatory postsynaptic potentials, meaning in this case, this rapid potential is we have our neurotransmitter open up a doorway. Now I've got a big attraction for sodium to come into the cell, so the electrical chemical gradient for sodium is much larger because I have two forces bringing it in, both the electrical, meaning it's attracted to the negative charge on the inside, and the chemical gradient, which again is the larger amount of sodium out here. Whereas my electrochemical gradient for potassium is much smaller. It is still going to move out, but I have one force trying to move it out. That was the chemical gradient going from high to low. And I have one force that was trying to hold it back, which was the electrical gradient, because it was being attracted to those negative charges inside of the cell. Therefore, I'm always going to have more sodium move through the same channel than potassium moving it out if it's a channel where both